Bismillahirrahmanirrahim. Today we will discuss production, breakdown and function of RBCs. Erythropoiesis, the production of red blood cell, is also called erythropoiesis, and it is an active process which maintains normal number of circulating blood cell and responds rapidly to increased bleeding or hypoxia. When there is low tension of oxygen or bleeding causes decreased number of RBC. So for normalization of the number of RBC, this process is activated. Or when there is low tension of oxygen, a production of RBC occur. Although normally this process is continued throughout the life. Haematopoiesis. It is also an active process, and the causative agents are infection or bleeding. When there is infection or bleeding, there is production of all blood cell. Haematopoiesis means production of all blood cell. All blood cell mean the production of red blood cell, white blood cell, and so bleeding in both cases is common here in erythropoiesis and also in haematopoiesis. But in haematopoiesis, all blood cells are produced. Haematopoiesis occur during in the early week of embryonic life, and primitive nucleated red blood cells are produced in the yolk sac. Production of red blood cell by bone marrow. During intrauterine life, development of the fetus in the womb of the mother, and when bones appear, the bone marrow is also developed in the bone marrow. Start production of RBC. Before the development of bone or bone marrow, this function is performed by the liver and spleen. After birth, up to the age of 5, all the bone of the body produce red blood cell. The bone marrow of all bones produce red blood cell. Here in this graph you can see that up to the age of 5 after birth, all the bone are producing red blood cell 100%. But with increase in the age, there is decline in the production of RBC by the long bone. Tibia stop production of bone marrow at the age of 20, and this brown color show that the femur stop formation of red blood cell at the age of 25. After the age of 25, production of red blood cell is then taken over by the spongy bone. At the age of 70, decline to the 25%. Production of RBC by is declined to 25%. The red line show the sternum. So the sternum also decline. After the age of 5, decline also occur in production of by the sternum, and at the age of 20, decrease up to 25%, and at the age of 70, it decreased to 45%. A child below 5 years of all his bone are involved in production of RBCs. After the age of 20, the long bones, they are become non-productive, they are not producing RBC, and this activity is taken over by the spongy bone like the ilium, the ribs, the sternum, clavicle, and the skull bone. Only the proximal portion of the long bone are responsible for production of red blood cell. In some cases, extramedullary production of RBC. Production of RBC outside the bone marrow. In some cases, the liver, thymus, and spleen may resume their haematopoietic function. This is called extramedullary haematopoiesis. It may be cause these organs to increase in size substantially. In fetal life, this is a physiological condition, and in adult life, it is a pathological condition. HSPCS, haematopoietic stem and progenitor cell. HPS cells, they are responsible for the production of RBC by the liver and spleen during fetal life, and when the bone and bone marrow develop, this function is also shaped into the bone marrow, and then the bone marrow start production of RBC and adding it into the circulation. Before the bone marrow, the liver and the spleen add the cell into the circulatory system. But some diseases such as myeloproliferative neoplasm, MPN, and lymphomas and leukemia, when the proper function of the marrow is deteriorated. This is pathological condition. In physiological condition, the production of RBC are shifted from the liver and spleen to the bone marrow. In pathological condition, when there is myelofibrosis, progression when the myelofibrosis, when the bone marrow is converted into fibrous tissue and they are no more capable of production of RBCs, again this process is shifted to the liver and spleen. This is a pathological condition and it occurs in some disease condition like leukemia or myelofibrosis. In this case, the bone marrow either occupied by the white blood cell or by the fibrous tissue. So in this condition, the liver and spleen take over the charge. Red marrow and yellow marrow. The bone marrow that actively produce blood cell is called red marrow. These are the red marrows which are producing the red blood cell. So they are called red marrow. And bone marrow that no longer produce blood cell is called yellow marrow. Multipotential haematopoietic stem cell or hemocytoblast. This cell is present in the bone marrow and it is responsible for the production of all type of cell. Stages of differentiation of red blood cell. How differentiation of red blood cell occur? The first cell that can be identified as belonging to the red blood cell series is the proerythroblast. This cell is produced from the multipotential haematopoietic stem cell. This is the first cell which occurs is proerythroblast. Under appropriate stimulation, these cells divide multi-time. Proerythroblast has been formed. This proerythroblast when formed, it divides multiple times, eventually forming many mature red blood cells. The first generation cells are called basophilic erythroblasts because they stand with basic dye. When these cells are passing through the staining process, they take basic dye. The cell at this time has accumulated very little hemoglobin. This basophilic erythroblast is then converted into polychromic erythroblast. This is the polychromic erythroblast because it stands with multiple polychromatophilic erythroblast. The other is orthochromic erythroblast. In this cell, the color is reduced. The multicolor colors are reduced to one color, so it is called orthochromic erythroblast. And in the next stage, the nucleus and the organelles removed from the cell or reabsorb. This cell is called reticulocyte because at this stage the cell is called reticulocyte because it has a mesh of the organelle of the cell, and when these organelles are totally absorbed at this stage by diapedesis, the cell is transferred from bone marrow into the bloodstream by diapedesis, and in the blood then the erythrocyte is mature. This is a mature erythrocyte by concave disc. Here you can see this is the proerythroblast, the basophilic erythroblast, and it is polychromatophilic erythroblast and orthochromatophilic erythroblast. The other thing that is initially when at this stage there is very little hemoglobin, but with the passage of time the concentration of hemoglobin is increasing and the RNA is decreasing. At the end, the RBC accumulate hemoglobin up to 34 gram. This is a mature erythrocyte which is finally developed from the proerythroblast or multipotential cell in the bone marrow. Senescence. Here you can see that this is a normal cell and this is senescent cell. This is a process in which actually this is the normal cell. We know that the RBC contains no nucleus, so it cannot make its copy, and that's why its lifespan is very short. It is 120 days. When it cannot make its copy and it's become old, the cell membrane is become wrinkled. This wrinkling make it susceptible to phagocytosis, a system which is present in the reticuloendothelial system, the spleen, liver and lymph node. The macrophages, they have the capability to identify these old wrinkled cells, and when they pass through this system, they capture them, remove these cells from the blood, and through this mechanism, this system continually purging or purifying the blood. The heme constituent of the hemoglobin are broken down into iron and biliverdin. Hemoglobin are broken down into iron and biliverdin, and then the biliverdin is reduced into bilirubin, which is then released into the plasma and transported to the liver, bound to albumin, and from liver it is shifted to the gallbladder, and from gallbladder by common bile duct it is reached the duodenum, and by gastrointestinal tract it is cleared from the body. The iron which is released into the plasma, and this iron is either recirculated or stored in the body. It is bound to a protein called transferrin. When it is needed for production of or erythropoiesis, red blood cell, it is directly transported to the bone marrow and reused for the production of RBC. If not needed, the iron is stored in the liver and